Thank you. Good morning, everyone. So I'm new to the University of Minnesota. And on the next slide, I'll give you some sort of background as to where I've come from. But to start, I know that I've been told by my American colleagues in Minnesota that people don't understand what bespoke means. So in England, bespoke means custom. So this idea of us creating a custom pipeline to sequence full genomes of PRRS. My background is in non-animal systems. So this is, again, a conference that's relatively new to me. I've worked on viruses infecting algae, and I've also worked on viruses infecting honeybees, and that's one of the main reasons why I'm in the US. I'm trying to understand some of the viruses infecting in honeybees. But I guess as to summarize where I am in terms of my lab within the context of the questions I'm asking, what I'm trying to do is really ask questions what viruses are doing to ensure that we can have a good, secure food supply. And my son loves his ham, and he also loves his honey-roasted ham. So I think there's a very good reason for working on pigs and honeybee health. So again, I'm not going to talk about the other projects I work on in terms of ASF or bovine leukemia virus, but I'm going to focus mainly on PRRS. The project I'm going to describe now is funded to the NC229 Project Award that I received last year. And I also collaborate with a variety of researchers, Monsieur Tomarel from the University of Minnesota, Cesar Koza, Kim van der Waal, all of those interested in PRRS and PRRS dynamics. And there's a project that was funded through the National Pork Board that gave us some samples to test our technique. The also advantage of moving from the UK to America is that means I had to start my lab from scratch, which also meant I had to get back into the lab. So I spent time last year developing protocols, and the postdoc, Jessica Cavill, was responsible for most of the data in terms of generating the raw data. And me, Yang, a technician in Monsieur Tomarel's lab, helped with the RNA extraction protocols. So this is why the ambition I had starting. Is that we wanted a diagnostic tool for researchers, but also for diagnostic labs like the VDL, to create a surveillance network so that we could actually sequence full genomes reliably and cheaper, and have it, as you heard this morning, accurate. And so this is the sort of strategy I set my sights on, in that the sample we wanted to work on was not a virus amplified sample. When isolate, we wanted to work on samples that came from the field, so serum, processing fluids, oral fluids, etc. We also wanted to use a high throughput system like the MagMax system. Okay. I'll go a little bit more detail into what the library preparation. We decided to use this smarter kit, and again, I'll go back to this in a minute. And the sequencing technology, the new rage is this Oxford nanopore. So we really wanted to see whether the Oxford nanopore MinION sequencer could work for us. And again, bioinformatics, we wanted something really simple, so we wanted to go around and use a commercial software. And we believe we can do this within 24 hours, and I think it's possible. And this was our goal. Okay, so again, at the heart of this technology is the smarter technology. And the smarter technology essentially utilizes this RNA, positive stranded RNA nature of PRRS, which essentially means that all you need to do is one reaction in terms of the reverse transcriptase for the virus detection. Right? You don't need two primers, you just need the one. And because we're using this Takara method, we can actually amplify the cDNA and then amplify it further using standard PCR. I just show this here because everybody knows this is a pretty daunting task, because PRRS is a highly variable virus. There's a lot of changes. And so designing a primer anywhere in here is not that easy or straightforward. Nonetheless, I'm always up for a challenge. I downloaded 431 full genomes of the NCBI database, and I was able to find a region that I believe we could design a primer for, which we wanted to test. And this primer will be able to pick up European and American strains or versions or species, apparently I've been told, of PRRS. I'm not going to spend time on that. What I was going to say is that this primer essentially is located at the 3' end of the genome, so it'll amplify the genome. And the kit we use, again, this Takara kit, requires small amounts of RNA. We need picograms to nanograms. So again, you can work on material serum, tissue, where you can get low amounts of RNA, and this can be used within this strategy. Again, the smarter kit is really nice, because what it does is all you need is, say, the one primer at the end, and when the smart polymerase extends, it adds a sequence to this first strand cDNA. And the kits have a primer that will be able to attach to the sequence. And if you do that, you can end up creating, through PCR, the selection of your primer. And in a very simple step of cleanup, you end up having a product ready for sequencing. This company has also developed a tool where they can add an additional barcode, so you can actually add an Illumina barcode into the system, and then when you add an Illumina barcode, there's an additional step for ribosomal cleanup, because you can start with total RNA, you don't have to work with a messenger RNA. And if you do that, you can deplete the ribosomal contaminants. Again, I'm not going to go into detail, but what you do end up getting is a selection, again after PCR cycling, using a primer that's you added on to the smarter linker. You can then amplify your signal. And of course, this technology means you can barcode, you can run more than one sample at any one time, and you can sequence using Illumina, or in our case, we can sequence using MinION. So this is all theory. What's the results? The results are that usually when you take a total RNA sample and you do whole genomic sequencing on it, you generally get less than 1% of your virus as being the virus in that material. You generally pick up the rest of the host. We've improved this now. We can get 40%, and the majority of this virus is PRRS. Now remember, this is a MinION sequencing output, so there's a lot of errors in it, so I don't read too much into some of these other viruses we're picking up, but it's something that we will be looking into and improving upon. And in fact, just last week, we were able to get 70% of all our reads being PRRS. So this is being improved all the time. So one of the samples we tested is a serum sample, and we used a CT of 25 because we wanted it to work, so we wanted to make sure we can do this. And this is a 2-hour run, right? In 2 hours, we were able to go from zero knowledge of this virus to a full genome, and really good coverage, right? And we get some really long reads, so assembly is really easy. And with unbelievable sequence depth, right? We were able to look at this sequence in depth, and we can on 5'N find all the markers, all the smarter. So in black is the smarter oligo, which the company wouldn't tell us, but now I know what it is. We have Illumina adapters wedged in between. We have these various primers. We can identify all these areas in terms of so we know the application's working as the protocol suggests. What we did notice as well is that some of the primers in the kit, in the mammalian kit, can actually amplify PRRS directly. It doesn't need this linker. And so we know there's off-target amplification, but that's something we think we'll be working on in the future to try and improve, and hence get PRRS-specific sequencing only and not to the host genome and other variants. And again, on the 3' end, we find the barcodes that we need. So every color here represents a different barcode. So we can run more than one sample at any one time. And again, we've validated this data. We know how and what sort of threshold we need to set to remove any non-specific, sequence error as based on MinION sequencing by comparing that to Illumina. And we know that the quality of the sequence, because of the depth we're getting, is as good as Illumina. Okay? And again, because of the sequence quality, we weren't sure that we could separate different virus isolates when you run it at the same time. And the answer is yes, we can. So, for example, we mixed the vaccine strain with a virus that we knew was contemporary, and using the sequence analysis, we were able to say, well, actually, yes, this isolate here is the vaccine strain, and this isolate here belongs to this closest Minnesota strain. So the sequence is good enough, just on a rough click of a button, to tell us which variant is in our pot. Now, just on close examination, we wanted to have a look at the sensitivity of this technique, and also using a real-world case. So here we have pigs, 4 pigs that was exposed to this virus, and we have controls where the pigs prior to the experiment, and then post-infection, 1 day through to 14, and these samples were taken, these are serum samples, and using both ORF5 and ORF6 amplification, we knew where the virus entered these pigs. And we chose samples that were negative, CT negatives, and those were really positive. What was interesting is that even on ORF5 and ORF6 qPCR primers, we have produced negative results. This one, these in the box. We were able to actually detect virus in these animals. So a CT negative, right? No detection on ORF5 and ORF6. This assay can detect this virus. Really surprising. We thought, this is crazy, this is impossible. But it works. And actually, when we look at the sequencing as a whole, it looks pretty good. And when you try and separate these barcodes out, we do know there's a 3' bias for some of these really weakly amplified samples. So there's an issue with those that are there in really low concentrations. We get either fragmentation through the pipeline, or the RNA is not in really good quality that the qPCR misses, but our technique picks up. We've also had a look at the SNP reads to these samples. And every line here tells you there's variation. And again, this is quite surprising. I work on a lot of viruses, and I'm really surprised how divergent and diverse, how rapidly this PRRS virus changes. Mike Murtaugh warned me, and he said, Declan, you'll never understand this virus as well. He's probably right. But what's interesting is that you can actually identify hotspots. So there are some regions in the genome where actually, depending on what animal you look at, these areas are consistently ambiguous. And I wanted to show you this slide because we were able to see, for example, in one pig. So this is exactly the same region of the genome for this pig 28 as opposed to the rest of the pigs in the study. And we mapped this genome relative to a reference genome that we know it's not the same. So the sequences that differ are shown up in color. But for example, in this pig 28, you have a base pair SNP that is consistent in pig 28 that's not found in the other pigs. What's also important is that you can have base pair changes even within a pig that are variable, and this is what the ends would be in Sanger sequencing or in Illumina. So actually, because we're doing strand sequencing in Oxford nanopore, we can separate those ends by individual bases. So now we can get a better resolution in terms of the variation in PRRS, and I feel that this technology allows us to better describe when this virus is mutating and what it's mutating to. So just to end off, we believe we've got a technique, and we've tested this. We can go from one sample from extraction to full genome within 24 hours. Okay? If you run multiple samples, it becomes more tricky because the bioinformatics becomes more labor intensive, and the MinION run takes longer to do base calling. So we can go minimum 24, a maximum 72 hours. Within 72 hours, we're able to do 72 samples, okay, full genomes. We know that there's a problem with the barcoding. So for the Illumina side of the barcoding, it means that we are not getting full length, and we know there's degradation parts through the process, and so we lose the Illumina barcodes through the process. So barcoding and increasing the yield and the numbers is not that easy, but barcoding using the MinION is still possible. And the real good news is that we believe, do I put a number here? Yeah, we believe we can get it down to in the 80s, less than $100 per sample within, let's say, within 72 hours. See if we can get the barcoding to work optimally. If you want a bespoke, really clean, really high accurate genome, it could be around about the $180, $200 mark. And actually, this virus, we believe, this technology can be used for other positive strand RNA viruses. I know I'm using it for my honeybee RNA virus. So I think this can go beyond just PRRS. It can go to the other positive strand RNA virus. And as I say, there's more to come. There's a lot of projects that are currently working on, as we speak, looking at different sample types. We're looking at tissues. We're looking at processing fluids. Results are tricky at the moment, but I believe it's doable. So watch the space. If anybody knows that anybody else is doing something similar to this, or you've seen this before, please let me know, because we've just filed a patent on this last week. I don't want to be wasting money and time. If someone else has done this, being new to the field. Thank you for your attention.
We have time to take one quick question.
Good morning. Thank you very much for your presentation. My question would be double. The first one is concerning the full genome coverage, which is the proportion of complete genomes that you obtain from a single read with the ACT4 nanopore? And the second would be if you have estimated the rate of error of your sequencing. Thank you very much.
Yeah, good question. So from an isolate, we can get 2/3. I haven't got a full genome just yet for PRRS. For my other viruses, I have single reads that go from start to end. Okay, so I know it's possible. For me, I got a feeling there's something to do with the structure of this virus that doesn't allow it to go through that easily to the end. But I do believe, I'm confident. I always think that the glass is half full. I think it's doable. Error rate, I don't know yet, because this is something we are continually trying to fix because of the min-iron problem. The min-iron produces lots of error. And so on an individual strand basis, you need a minimum, in region basis, you need a minimum of 10-fold coverage to get anything which is reasonable. Okay, so, but in terms of rate and figures, no, I don't have that for you just yet because we are currently working on that.
Thank you very much.